Dear students, welcome to another class of biology. On the previous day, we had started the chapter on genetics and we will continue with that lesson. Probably we are going to complete it also today. So on the previous day, I have told you the different conditions which led to the choice of the garden pea as the experimental substance by Mendel. And I have also told you the criteria for carrying out the cross, monohybrid as well as dihybrid cross, and we have done some crosses as well. I have given some as homework also. Hope you have done it. So now we are going to continue with that. So we have completed monohybrid cross, we have completed dihybrid cross, and today we are going to do sex determination. So before we come to sex determination, there are two terms which should be made clear to you people. That is, one is the test cross and the other one is the back cross. Quite often they ask for the definition of the test cross and the back cross. Now when we were doing the monohybrid cross, say we have taken a tall plant and we have crossed it with a dwarf plant, and then in the F1 hybrid we got capital T and small t. This cross I have already done, and all these plants were found to be heterozygous tall. Now if we take the F1 hybrid and if we cross it with any of the parent, instead of crossing it with another offspring of F1 generation, then that becomes a back cross. Now what is the meaning of that? Say capital T, try to recall the thing which you have done before. Small t, this you have done. Tall is getting crossed with dwarf. You are getting the gamete formation. So T, again small t. In F1 generation, this will again combine, okay? And in all the cases, you will be getting capital T and small t. This I have already done. But when we are working out the F2 generation, we have taken two hybrids from the F1. That means for the F2 generation, we have to cross the hybrids like this, isn't it? But instead of doing that, if we take the F1 hybrid and if we cross it either with the dominant parent or the F1 hybrid is crossed with the recessive parent, then that becomes a back cross, BACK, back cross. That means we are going back to the parental generation. What we are doing? We have taken one hybrid from the F1 generation, and instead of crossing it with another hybrid to get the F2 generation, we are crossing it either with the dominant parent, that is capital T, or we are crossing it with the recessive parent, that is small t. Then that becomes an example of a back cross. And another term is there, that is test cross. So what is test cross? When the F1 hybrid is crossed only with the recessive parent, only with small t or only with the recessive parent, then that becomes a test cross. Try to get the difference. When the F1 hybrid is crossed either with the dominant parent or with the recessive parent, then that becomes a back cross. And when the F1 hybrid is crossed only with the recessive parent, then that becomes a test cross. So we can say that the test cross is a part of the back cross. Means we can say all test crosses are back crosses, but all back crosses are not test crosses. Because if it is a test cross, it has to get crossed only with the recessive parent. But if it is a back cross, then either the recessive parent or with the dominant parent. Okay, so all test crosses can be called the back crosses, but all the back crosses are not test crosses. Okay. So they might ask you to explain it, or they might ask you the definition, or they might ask that why all test crosses are back crosses. Explain, that way they can give the question. Usually they will ask you to write the definition or explain the term. In that case, you can pick up any example, like this example will also do, and you have to explain it in the way that I have done. Okay, so this concept should be clear. Another one is incomplete dominance. What is that? Incomplete dominance. Incomplete dominance. In this cross, we will see that when the condition is heterozygous, then it becomes intermediate between the particular character gets intermediate between the dominant character and the recessive character. I can explain with an example. Like, let us take red flowers and we cross it with white flowers, and we all know that a plant having red flowers is crossed with a plant having white flowers. We all know that red is a dominant character and that is represented by capital R, and white is a recessive character which is represented by small r. Okay, so the same way as we have done the cross, capital R, small r, this is the red flower and this is the white flower. As usual, gamete formation, so we will be getting the gametes like this. So R will separate out, the small r will also separate out in the form of gametes. Now for the F1 generation, what do we get? We will cross it like this, the same type of cross, monohybrid cross, and in each case we will be getting capital R, small r this way. Okay, so we will be getting 4 F1 generation. Now when we have done the normal this monohybrid cross, then what did we find? It is also a type of monohybrid cross, isn't it, when we are taking one pair of contrasting characters. So here red, when it is combined or rather crossed with a plant having white flowers, then that is producing flowers in the F1 generation which are all red in color. But what type of red? I told you before, it is heterozygous. It is not pure or homozygous, it is called heterozygous because there is one capital R and one small r. Now in this case, in the case of incomplete dominance, you will find that these flowers, they are heterozygous no doubt because one capital R and one small r, but the color is going to be pink, not red. Why? Because in this case, this dominant character will be expressed only when it is homozygous. That means when there is capital R, 2 capital R's when we get, when that is when it is in a homozygous state or pure state, then only this red color or the dominant character will be expressed. But if it is in the heterozygous state, then you will get an intermediate condition, neither red nor white, something in between, that is pink. That is known as incomplete dominance. Okay, so when in the heterozygous condition we get some intermediate character, then that becomes an example of incomplete dominance. But the way you work it out will be the same as you work out a monohybrid cross. So if you are supposed to continue with this, what we do right here, heterozygous, but they are pink flowers. So they will produce pink flowers. This is the F1 generation. Now we will go to the F2 generation. We will take 2 plants, each one is capital R and small r. Capital R, small r, cross capital R, small r. These are the products of the F1 generation. Again we will do gamete formation. So it's capital R, small r, again separating out capital R, small r. Then we go to F2 generation. Let's come to the Punnett square. Okay, so we go to the Punnett square. So we put the symbol for the male, we put the symbol for the female, and we are going to write down the gamete. So this one, capital R, small r, if we take this as the male plant, then these 2 are the gametes of the male plant. So capital R, small r, and these 2 are the gametes of the female plant. So we are writing these 2 gametes along this line. That is, this represents, the symbol represents the female. So along this we are going to write it. Okay. Now, when we combine them, what do we get? Capital R, capital R, capital R, small r, capital R, small r, small r, small r. Then as usual, we have to find out the phenotypic ratio. That means the ratio which is based on the observable characteristics or the external features of an individual. So that is called the phenotypic ratio. So what will be the phenotypic ratio? This one is a red plant, I should not say red plant, a plant with red flowers. This is a plant with red flowers, isn't it? What color will be this? This will be pink. I told you it is incomplete dominance, so it is going to be, the condition is going to be intermediate between the dominant and the recessive. So it will neither be red nor will it be white, it will be intermediate between the two. So it's going to be pink in nature. So this one will be red, this pink, this one also pink, and this one will be white. So what will be the phenotypic ratio? If you just go by the color, this is red, these two are pink, and this one is white. So this ratio becomes 1:2:1. See how it differs from the original phenotypic ratio that we get in the monohybrid cross. In monohybrid cross, we get the ratio as, the phenotypic ratio as 3:1. And the genotypic ratio is 1:2:1. Here we find that the phenotypic ratio has also become 1:2:1. It has changed because of incomplete dominance. Okay. And what will be the genotypic ratio? The ratio that is based on the genetic constitution. Genotypic ratio. This one is homozygous red. These two are heterozygous pink. This one is white. Okay, so this will be again 1:2:1. So in this type of cross, you find that the phenotypic ratio and the genotypic ratio are the same. So if they ask you what is the deviation from the monohybrid cross by Mendel, then you can give the example of incomplete dominance, where in the heterozygous state you get an intermediate color, okay, or intermediate characteristic. It may be color, it can be any other thing. So you get an intermediate characteristic, that is incomplete dominance, okay. And another point is there is codominance. You can just remember the name codominance, where both the characters are dominant. You don't get any recessive form, okay. They both are dominant. So that is the meaning of codominance. Co means at the same time, two dominant characters are existing at the same time. So it is codominance. So in the case of codominance and in the case of incomplete dominance, you will find that the phenotypic ratio is going to change. Okay. Now we will come to sex determination. We'll start with the human beings. Now when we go to the, the classification of the chromosomes rather, we will have two main categories under which we can put the chromosomes. One is the body chromosome, which is known as the autosomes, and the other one is the allosome or the sex chromosomes. So if you take the human beings, in human beings the chromosome number is 46. That means there will be, and there are two sex chromosomes. So if the chromosome number of human beings, chromosome number in human beings it is 46, okay. And the male as well as the female, they have got two sex chromosomes, okay. So in the case of the females, you can represent by 44 plus XX. 44, these are the body chromosomes, or we can call them autosomes, and these two are the sex chromosomes, which we can call allosomes. Got it? So in this case you find that both the sex chromosomes are the same. That means two X chromosomes in the case of the female. So 44, 45, 46. Total number is 46. Next, in the case of males, you will find again the autosome number will be 44 as usual, and two sex chromosome in order to become 46. So one is X chromosome and the other is Y chromosome. So you can see that this heterogametic, that means if there is one X chromosome and one Y chromosome, these two are different, whereas here we are getting the same type of sex chromosome, that is XX. Okay, so when we represent a male, we represent it by 44, which is the autosome number of body chromosomes, plus XY. So 44, 45, 46, total chromosome number becomes 46. Now quite often they ask you the question that how can you explain, or how can you say that the sex of a child depends upon the father and not upon the mother. For that I need to do a cross where we will not take the autosomes into consideration because they are the same in both. We will just take the sex chromosomes into consideration. The male is getting crossed with the female. So this one is represented by XY, as I've told you, these are the XX chromosomes, and this one is represented by XX. We are not taking the autosomes into consideration. Now at the time of gamete formation, what's going to happen? These two will separate out X and Y, and these two will also separate out X and X. Now if the X of the male combines with the X of the female, it forms XX. If the X of the male combines with the other X of the female, then that also is XX. If the Y of the male combines with the X of the female, it becomes XY, and again here also we will be getting XY. This is going to be a female child, this is going to be a female child, this is going to be a male child, male child. Okay, so it's 50-50 probability. Now here you see that, now how can we say that the sex of a child actually depends upon the father and not upon the mother? Because in the females, you see, they have got both the sex chromosomes of the same type. In their gametes, you will see this is one gamete coming out from the female, another gamete. So the same type of sex chromosome is coming in the gamete. So we can say they are homogametic. The gametes which are being produced by the female are of the same type. Each of the gametes is having one X chromosome. That means the female is homogametic, producing the same type of gametes. Whereas in the case of the males, you find if one gamete contains X, the other gamete contains Y. So the male is actually heterogametic. It's different gametes are being formed. So it's heterogametic. Okay. So if the X of the male combines with the X of the female, then a female child is formed. If the Y of the male combines with the X of the female, then a male child is formed. So you can see that the female does not have that particular sex chromosome which is able to form a male child. So quite often in a family you will find that the females are actually, they are rebuked and they are held responsible, they are blamed for not being producing a male child. But if you go by genetics, a female doesn't have the capacity of producing the male child because it has got the same type of chromosomes. So it has got both X, it doesn't have the Y in it. Then how can the female produce a male child? So it depends upon the father. The sex of the child actually depends upon the father, because if only the Y of the male is able to combine with the X of the female, then a male child will be formed. So the capacity of forming a male child by donating the Y chromosome is there with the male only. The females can only produce two gametes, each having one X chromosome. Okay, so for this reason you have to explain this, and for this reason we can say that the sex of the child depends upon the father and not on the mother, because the males are heterogametic whereas the females are homogametic. Now next we will come to the sex-linked disorders. Sex-linked disorders. Or diseases. Now, say for example, let's take the Y-linked diseases. Y-linked diseases. We don't have to work out any cross in this. Like if there is any mutation or any change brought about in the Y chromosome, then it can lead to some disease. You know that there are genes on the chromosome and each of the gene is responsible for performing a particular function. Now if we find that there is a mutation or some change in the gene, then that might also produce some different characteristics which we can call disorders. Okay. Now if this type of changes occur in the genes of the Y chromosome, then we can call those diseases Y-linked diseases. And this will be found only in the case of the males, because you know the females do not have Y chromosome inside them. So just remember the names of two diseases. You don't have to work out any cross here. So one is hypertrichory, and the other one is baldness, where there is no hair on the skull, isn't it? Baldness. So these two are actually examples of the Y-linked diseases. Then we will come to the X-linked diseases. X-linked diseases, now which are common in both the males and the females. One is color blindness and the other one is hemophilia. hemophilia. Color blindness is a condition where different shades of say blue, then different shades of red, these things cannot be actually identified. And hemophilia, where the clotting factor is absent, and whenever there is a cut or wound in the body, a lot of bleeding takes place. So these are the X-linked diseases, color blindness and hemophilia. And in the case of the Y-linked diseases, you have got two examples. One is hypertrichory, where a large number of hairs will come out from the earlobes, etc., and baldness, absence of hair, that you know very well. So these two are linked with the Y chromosome, and these two are linked with the X chromosome. Since these two are linked with the X chromosome, it can occur in the males as well as in the females. Now we have to do some cross on this color blindness, the same type of cross for hemophilia also, because they fall under the same category, but we can do that on the next day. We'll continue on the next day. Okay, so much for today. Thank you.